Hi, everyone, and welcome to our Advocates webinar for Modernizing Oncology Endpoints. I'm Hillary Andrews, Senior Director of Science Policy and Strategy here at Friends. Thank you so much for joining us. A few housekeeping notes before we get started. We'll ask that you ask questions using the question and answer box on Zoom. We're recording the event today and it will be posted later on YouTube. We have a great panel for you, but before we get started, hopefully you were able to review some of our materials from the event that we had on February 5th, but we wanted to give you a little bit more background. Biomarkers are measurable indicators of biological processes or states. We use tests or assays to measure or understand biomarkers, and there are a variety of different biomarkers as outlined on this slide, which provides some information about the NIH FDA best categories. We have diagnostic biomarkers, predictive biomarkers, and safety biomarkers. But we also have response biomarkers, which is the focus of our discussion today. They're used to show a biological response has occurred in an individual exposed to a medical product. That can be for pharmacodynamic, but here we're focusing on early or surrogate endpoint biomarkers. To understand how early endpoints can help us in clinical trials, we want to first go through a clinical trial timeline. Multiple patients are enrolled on a clinical trial and begin treatment, and after a period of time, a clinical endpoint such as overall survival is measured. A drug approval decision is made by FDA based off of information provided in that clinical endpoint. Early endpoints provide information on whether a treatment is benefiting patients sooner and can expedite evidence generation needed for regulatory approval of therapies. We have early endpoints in oncology that are already in use. RECIST is a set of criteria that are used to assess tumor change over time on tumor images from CT scans. They define endpoints such as objective response rate, ORR, or progression-free survival, PFS. It has provided value for over 30 years. However, there are some limitations. Choosing which tumors to measure is manual and can be subjective, which may make it harder to track how cancer changes over time. Measuring tumors in just one direction may miss treatment effects that full 3D measurements could catch. These methods can't always tell whether a tumor has grown because of treatment effects, such as pseudoprogression, or because the tumor is actually growing. And focusing on only the 5 largest tumors assumes they represent what's happening in the whole body, which isn't always true. Emerging approaches of biomarkers that could be considered as novel endpoints include circulating tumor DNA or ctDNA, which is isolated from the blood. Levels of ctDNA in the blood correlate with tumor burden, and monitoring changes in ctDNA over time can help to understand treatment efficacy. Another approach is AI-enabled tumor assessment, where AI-enabled tumor assessment tools analyze tumor scans. They can measure features in tumors that are imperceptible to humans. They can also measure RECIST and may do so in a way that's either better or more accurate than human readers. And these features that are imperceptible may help us to understand treatment efficacy. Using these approaches to develop novel early endpoints may provide earlier readouts or results that have stronger associations with outcomes compared to RECIST-based endpoints, but we need data. And so Friends projects help to develop that necessary data. The CT Monitor project asked the question, do reductions in ctDNA levels post-treatment correspond with therapeutic outcomes? Here we aggregated data from over 20 clinical trials and demonstrated that decreases in ctDNA levels are associated with improved outcomes. For our AI RECIST project, we're asking whether AI-enabled imaging tools can improve tumor assessments and accelerate clinical trials. We'll be sharing clinical trial images with 8 AI-enabled tool developers, and then we're comparing their RECIST outcomes to understand the level of agreement among tools, which will help us to understand whether these tools are ready to be included in clinical trials yet. And now I want to pass it over to our panelists, Naveen Zarifa, who's a principal and founder at NMD Group, Minetta Liu, Chief Medical Officer of Oncology at Natera, and Lauren Brady, Director of Translational Data Science from Genmab. Thank you to our panelists, and thank you all for joining us today.
Hillary, thank you so much for that summary. It was really clear and wonderful to follow along. And of course, thank you for all your work and that of the rest of the team at Friends. Minetta, Lauren, I'm delighted to be with you for the rest of the hour. I know that we have some conversation topics. We'll be getting questions from the audience as we go through. And just to say to the members of the audience, thank you for all the work that you do on behalf of patients through your engagement with advocacy. So we're here for you today. So as you were listening to Hillary and the work that we have been undertaking together in collaboration, could I ask you the big summary question? Why are early endpoints important in your mind? And I'm going to start with you, Minetta.
Sure. Thanks, Naveen. It's a pleasure to be here today. Early endpoints, to be clear, we're talking about endpoints for clinical trials. And we are very much in a world where we want to streamline progress. Having earlier, and one could argue more reliable endpoints than standard imaging as it's done today, has the potential to reduce the number of patients that are needed to participate in clinical trials to get to the answers more quickly. I would rather treat a patient with a shorter course of treatment and then abandon ship, if you will, if the drugs weren't working, to then be able to move on to either a different patient population with the same drugs or different drugs if we're focused on therapeutic trials. And all of this is an effort to improve the standard of care ultimately.
Yeah, that's fantastic. I think I heard their speed so that we know if from clinical trials, if a drug is going to hold promise. And then I also heard, well, okay, if we have enough evidence to know that there's an early marker to abandon ship and go to maybe something that has the potential to be more promising, we don't want to wait to do that. We'd like to go ahead and do that quickly. So thank you. Thank you for that. And Lauren, your thoughts. Why are early
Thank you.
points relevant?
Sure. Thanks, Naveen. And I can pick up where Minetta left off and add that, even in the case where we're still building that evidence generation and the endpoints haven't necessarily been approved by regulatory authorities, data from ctDNA and other sources like, AI-enabled RESIST could potentially help us make earlier drug development decisions, which also benefit the patient. So even in that event, we are happy to still share those data with the regulatory authorities and help bridge that gap into bringing our decisions earlier and faster to patients.
That's an important point as well as we look at all of the promising new medications or molecules out there. If we can figure out which ones are the ones that are going to work faster, then we can focus our energies there and maybe abandon the ones that are less likely to be productive on behalf of patients. So that impact on the drug development throughput, if you want to call it that, is also something to keep an eye on. Thank you, Lauren.
Can I maybe just add to that? I mean, when we think about drug development, Lauren, you brought up a great point. We go through multiple phases of trials, right? And each phase, sometimes you have to get to the phase 3 trials, the big randomized studies, thousands of patients. And in the end, it's not a positive trial. So if we have, again, reliable early endpoints as early as phase 1, that will help just the overall investment, time, energy, finances, et cetera, on the path to drug development and approvals.
Yeah, no, that's an excellent point. Thank you. So we're hitting on the question of like who's involved in the apparatus. And I'm thinking of the stakeholders. We've got patients, then we've got clinicians, and we've got regulators. So if we think of each one of those, and you can choose to address your remarks in any or all, what do these better early endpoints mean for each of the stakeholders? Patients, regulators, clinicians. Who would like to start? Minetta?
Yeah, I can start. It goes back to what we were just discussing. I think it is for patients, clinicians, the regulators, the developers, anyone who might be involved in the conduct, right, the development conduct of these studies. It's all about finding the right patients for the right study and the right drugs for the study, and the time in all of this. So it just, again, from my perspective, comes down to resources and getting to the answer more quickly, and perhaps more efficiently. We do a great job. I mean, the advances in care for cancer patients have been remarkable. They're not fast enough, right? And so who has time for that? So I think everything that we're trying to do with the early endpoints is really trying to answer that question.
I'm hearing a sense of impatience, and what we have is already better, but we can do better and better. So I love that. Thank you. And Lauren, your thoughts on this?
Sure, I can add with the AI Resist project specifically. Of course, we're considering all three, but I think we spend a lot of time considering the physicians actually, because Resist is it's a great tool that we can use to standardize and, accelerate across clinical trials, but it's a little bit laborious and cumbersome. And we talk a lot about what is the variation between readers, what is the variation between these sites. And if we had a way to improve that, to speed up the process, to take some of that burden off of multiple different radiologists reading these images for the clinical trials, we could certainly accelerate the timelines of our clinical trials.
Totally. And I heard precision there, and I heard reliability and efficiency was thrown in. Yeah, totally. Do you think I mean, this is a stray question just comes to mind. Do you think that all three of the stakeholders are looking for the same thing, which is reliable early understanding? I think I heard efficiency in there. It strikes me that that's got to be an important component for all of them, right?
But they'll all have a different lens, right? And
Mhm.
perhaps prioritization amongst those different qualities. The regulatory groups have a very difficult job because they will hold the key as to whether or not the early endpoints that are created are in fact reliable enough to accelerate drug development. Right? We don't want to lose an opportunity for something to move forward either, right? That's the flip side as we keep talking about efficiency, et cetera. Again, I think it's probably the lens. These are all important for everybody, but for the responsibility that each of those groups hold within the construct of clinical trials and clinical care overall, they will look at these different factors differently.
So regulators are likely to be a bit more cautious.
Absolutely.
And yeah. Okay, so let's get concrete just for a second. What did we learn from CT Monitor, Minetta? And then Lauren, I'm going to ask you, what do we hope to learn from AI Resist? So let's start with what we learned in CT Monitor.
Sure. CT Monitor was a remarkable effort bringing stakeholders from all walks of life really around ctDNA related clinical trials, where we have a lot of evidence in small studies with specific assays that suggest that ctDNA correlates with outcomes. But this was carefully looking at ctDNA with different assays, although we had certain metrics for which could be included, and groups that were willing to share their information in aggregate so that we would have bigger numbers and be able to more definitively show whether ctDNA was worth it really to take forward as an early endpoint. The focus was on non-small cell lung cancer just because of the number of trials that were performed. There were basically two series of analyses that were done with different combinations of drugs, if you will, trying to stay within certain drug types again for uniformity of response. And the bottom line is looking at CT ctDNA and measuring it over the course of time relative to treatment on a clinical trial showed that either clearing the ctDNA, meaning you see it and then you don't, or a decrease in levels, so it's still there, but a lot less during the course of treatment, did correlate ultimately with survival. So it speaks to the reliability, if you will, of ctDNA as a potential early endpoint for clinical studies. So this will help us to move forward. Again, I hope as a community to really bring this home and to incorporate this as an early endpoint in clinical studies.
What a lovely summary, and just reminding ourselves that there was a lot of interest in the scientific community, but everybody was pursuing it on their own. And what the project did was bring the data together so we could, in this collaborative setting, understand, is there really promise here? And I think you're telling us that, yes, there's reason to be hopeful that there's promise. So we'll talk towards the end. I'm going to ask you a little later, what's it going to take for this promise to really bring around concrete change? So I'll plant that seed with you and come back a little later. And Lauren, what are your views? Have we learned anything so far in AI.resist? And what are we going to be hearing from that project team in the next year or so?
Sure. I think one of the greatest values of this project is really bringing together the radiologists, the clinical thought leaders, and the AI developers and the researchers within the same room. And our conversations are always so lively. And we talk a lot about the promise of all of these exciting advances in AI biomarkers and all the things we really want to do. But we all stay grounded and aligned that, let's start with resist. Let's start by, making improvements to the thing that is already in place and showing people if they can adopt these techniques that could lead to reduced variability and increased precision, that we can improve what we're using already, and then we can build upon it from there. So it's been very exciting to really design these questions very thoughtfully with all of the experts in the room and trying to pull from the right data sets so that we can do some more advanced experimentation later on. So it's been quite exciting so far.
Wow. So both of them are large collaborative efforts. Both of them are saying, let's look at new approaches. One applied to something we know and have been working with for 30 years or so, resist. The other one is a whole new technology. So if you reflect back, is there some message, like a single message you can give the audience in terms of, so how does this represent a step forward from where we have been, these two projects?
I can try to
Or
take that one.
Okay, go for it, Lauren.
I think we talk a lot about evidence generation, and recently this is an exciting time for minimal residual disease and blood cancer, where the field, just in the same approach, kind of took this consortia-based approach and generated that data that they needed for more than 10 years to bring minimal residual disease to the stage of being included as a primary endpoint now recently. So I think that is what we're really aiming to do, but not take 10 years to do it, hopefully. And so if we can generate the right data, if we have the right experts in the room, if we have the right contributors bringing the data that we need to address these questions, maybe we can bring it, to use as an endpoint sooner.
Thank you. Minetta, anything to add?
no, it's an exciting time. And I think that we, with CT Monitor, again, in aggregate, agnostic to specific assays, et cetera, pulling this together just shows that we're really at the cusp. So the big question is, what do we do next to prove the point?
Okay, so we're going to come back to that. Let me bring it back to the patients for a second, and also patients within a trial. And I wonder if you could share your thoughts on how early and reliable endpoints, how that improve the experience of a patient within a clinical trial. What'd you think?
Do you want me to?
Sure.
So for a patient involved in a clinical study, treatment is ongoing, essentially in a clinical trial until it's proven to not be working, right? For that individual patient, if we're thinking about it at that level. And you can imagine for any patient getting treatment for cancer, why would you want to continue with therapy if you knew it wasn't going to work? You'd be incurring toxicity, side effects from the medications, and perhaps losing time, right? Because the whole goal is to be on effective therapy for as long as you need it and to stop it and maybe be free of cancer, right? And the whole goal with treatment in early or even later stage disease is really trying to get towards cure. Uh, so it's about, again, I'll go back to what we were talking about before. It's really saving time and energy and not continuing on a course that isn't going to work. And this also is why we need to be reliable, right? Because there are patients who have stable disease and continue on study and they are benefiting. Stable disease meaning nothing is changing radiographically or on the scans. But there's some where even things look fine on the scans, it's not going fine at a deeper level. And whether AI imaging is going to help us better understand what we're seeing on the scans or ctDNA at a molecular level will be more reflective. It's with the mindset of making sure the patient's on the right course of treatment.
So not only is there benefit in terms of speeding up the throughput for molecules being evaluated, there's also a benefit for the individual patient within the context of a trial. So that's an important point. Lauren.
Sure, I can add to that. I think with both the ctDNA and the imaging biomarkers, Minetta hinted at this, but we're getting a deeper, more nuanced understanding of the response and also the timing of the response, right? And so I think that, again, back to the evidence generation, the more that we can correlate some of these endpoints with biologically meaningful changes in disease, then that really helps move the bar in adoption of these endpoints and in our understanding of them.
We got a question from the audience that I'm going to pivot to, and it's a ctDNA question, something we covered off during the meeting, but we want to cover off here, and it will lead us into, okay, what more work do we need? So I'm going to take the prerogative here. I'm going to read it as it's written because it's very well written. So it sounds like ctDNA clearance predicts patient-level OS. Is there also a trial effect? In other words, where you're seeing a correlation, now I'm paraphrasing, seeing a direct correlation between the difference in two arms of an RCT for ctDNA clearance that totally lines up with the differences in OS. So what do we know right now in terms of the patient level versus the trial level evidence that Lawrence talked about for ctDNA? And I don't know, Minetta, if you want to cover it off or I'm comfortable doing it. Whichever.
I'm happy to start, and I'm sure you will embellish. So yes, at a patient level, ctDNA clearance is associated with better outcomes, both progression-free, disease-free, overall survival, depending on the study and the circumstance. The question is, even for an individual patient, does the degree of change, if you will, correlate with more of a benefit? And then on the trial level, perhaps more importantly, what do these changes mean for correlations with survival, et cetera? Direct observations with ctDNA at the patient level, the sort of slope of change, if you will. So it's not just how high you start and how low you go, but how quickly you get there can be relevant in terms of outcomes. Because unfortunately, when you look at one spot in time, things change over the course of time. So you want to look at the persistence of benefit or the duration of benefit as well. On a trial level, I think we're starting to learn. So when we look at retrospective studies where the trial is done, specimens were stored, and we're doing the ctDNA analyses, we can see that even if they're was a survival benefit. The trial was a positive trial, that we can make a trial more positive, or in fact, we can make a negative trial positive if you focus on the right population of patients relative to ctDNA. In other words, there have been clinical trials where there was treatment randomization. It did not show a benefit with drug, combination A and B. But if you focused on the ctDNA positive patients when they entered the trial, those are the patients you could argue needed help the most. And those were the patients that benefited from the newer therapy, if you will, the escalation of treatment. Those are more binary results, positive or negative at the beginning. And then if there are samples over the course of time and we're looking at the rate of clearance or the degree of clearance, we are starting to see hints that again, sort of that slope is making a difference. Naveen will describe this more eloquently in statistical terms, but just from what we see overall with trials, where you start will matter, and when and where you finish will matter as well. And that may have bearing on how much benefit we will see in overall survival overall, and whether that's going to change how we read out clinical trials will remain to be seen as we conduct these prospectively.
Yeah. So there are study designs out there that are now focusing on patients with elevated ctDNA early and trying to get to a better intervention for them, since we know that that's something that we can do today. But we were talking at the very, very beginning of our panel, the regulators and what they look for before they'll say, hey, I will put this drug on the market because I'm confident that it has efficacy for patients. They do look for the patient level associations that we've talked about, and then they look for that randomized controlled trial evidence across many, many randomized controlled trials showing that the differential between the two arms of a trial roughly line up, whether it's OS relative to ctDNA or PFS relative to ctDNA. And so to answer the question that was asked, at the trial level, what we've seen to date in CT Monitor, and its very early exploratory results, is that we did not see that nice lineup between ctDNA and survival. We think to some degree it has to do with the fact that patients can cross over to the active investigational arm after PFS. So when we look at the relationship between PFS and circulating tumor DNA, it's actually quite strong at the trial level. So again, a learning that we have to take into the future. And I think that's the last question we want to ask the panel today before we go to more questions from the audience. What does the future hold? What are the immediate next steps, Lauren, for AI RECIST? Because we absolutely want to get to change. So what in your mind do we have to tackle for AI RECIST?
Sure. So I think Hillary did a nice job summarizing our study design so far. And we designed this study to assess a comparison of 8 different AI tools, which is really interesting because we want to understand where that variability in the reader data is coming from. Is it coming from a human, or is it coming from the AI component, or is it coming from some outside, trial variation as well? And so that is really a very critical part of our study design. And one thing we talk about a lot is that the public almost oversimplifies this concept of AI data versus human-generated data, when in reality, it's actually a collaboration of the human physicians using the AI tool. So it's almost like AI plus human versus human alone. And so it will be really interesting to see all of these different comparisons and how they line up with each other, and whether that can help us, lead to earlier adoption of some of these tools in the clinical setting.
So the humans aren't going anywhere. They might
Humans
have
aren't going, yeah.
tools
We're still
to
here.
make their assessments, but we're not ready to let them go.
Yes, we still love our radiologists. Yeah, we don't want them to leave.
Okay. And And do you have a general idea when we might see results from AI.resist? What do you think? Is it going to take a couple
I
of?
think we're still in the process of sourcing some of the data, and so hopefully later this year we'll come back with an update, or maybe early next year.
Okay, that'll be great. Thank you. Minetta, what do you think for circulating tumor DNA? There's a very big set of options for us in terms of the future, but what are your priorities there?
So I think one important thing, just and maybe it's already understood, but I do want to point it out for ctDNA. What we're talking about here is ctDNA as a biomarker for minimal or molecular residual disease. So finding cancer, if present, as early as possible, that it's there, even if you can't see it on a scan. We're not talking about using a blood test to identify a specific target for a drug for drug selection. And that's a nuance, but I just do want to point that out. So as we're focusing on ctDNA or circulating tumor DNA for molecular residual disease, it can be placed in many places for clinical trials and for patient care. But here, as we're focusing on using it as an early endpoint, it's designing the trials in the right way to answer the point number one, and that's clinical trial design 101. But I think pertinent here, it's advocating for participation in those clinical trials. We need to generate this data. We spoke about evidence generation, and although these tests are out there and they can be used and they're starting to get into guidelines, the way we are really going to accelerate patient care, which is what we are all focused on, is supporting the clinical studies that are going to generate the data to get this solidly number one, this meaning MRD detection and guidelines, but importantly, enable its adoption as an early endpoint for drug development. So again, it's thinking about the right studies and participating in those right studies. Everything gets lumped together. I think human behavior is lumping. So we have lots of ctDNA trials, and they're all going to answer the question, but they're going to answer different ones. And it is critical for us to focus on establishing this as an early biomarker for inclusion as an endpoint in trials.
Very good. And I wonder your thoughts, the two of you, on where patient advocates play a role here in terms of getting us to the next big evidence generation components for this and other early endpoint options. And maybe we'll get thoughts from the audience as well as we go through, but what's your initial thoughts on the role of patient advocacy? Is it communication, translating the science? Is it the call to action, Minetta, that you just put out in terms of RCTs and patient participation there? Where's the biggest payoff, do we think?
Patient advocates are critical to making sure that clinical trials are designed with the patient in mind. So their support and involvement in clinical trial development. And then when there are established studies, it is education, right? It is sort of the call to arms and spreading the word. There are a lot of patients who have or ctDNA again is being used, right, in clinical practice in some places. And I'm not saying it's wrong or right? But again, to support the use of ctDNA as an endpoint in studies, we have to do those studies. So if a trial is available and if a patient is eligible and if a patient is able to participate, right? Because there's a matching process. Not every trial is not right for every patient or vice versa. But it's optimizing the ability for that interaction to happen.
Perfect. And Lauren, your thoughts, early thoughts on where patient advocates play a role?
Yeah, Naveen, I liked what you said about translating the science in addition to everything that Minetta has listed out so nicely. I really like that role, and I frequently have calls from sites and patient advocates saying, "Hey, what is this AI biomarker in the protocol? Why are we collecting this? My patients are concerned. What does this mean?" And we're happy to take those calls and talk about the science and why it's impactful and why we need to start collecting these data to bridge some of these concepts like ctDNA and AI resist. And so I think it's really great to have that partnership with the patient advocacy teams to help interface those interactions.
Great. And that leads us to questions from the audience. So if we're looking for them to help with the translation of the science, we'd better make sure that we are there helping with the questions that they have about the science. So we have a question here, and it's about ctDNA. Are there certain cancers that lend themselves better in using ctDNA analysis? For example, do brain tumors actually provide ctDNA in the bloodstream? So let's take the first question of like, what do we know across different tumor types, Minetta? And then talk about the blood-brain barrier and brain tumors.
So different tumors will shed, if you want to call it shedding, or putting out ctDNA into the bloodstream at different rates, perhaps, or it has to do with the circulation itself. So there are correlations with the ability to detect ctDNA in the bloodstream relative to cancer type. It has to do with sometimes with where, again, where the cancer is, the tumor is located and how the blood circulation is around that area. It could be the rate at which a tumor is dividing and, or we call it proliferating and growing. So there are many factors that I think we're just starting to understand the biology, if you will, of ctDNA leaving a tumor and going into the bloodstream. There are also factors for ctDNA trying to get into the bloodstream and then local factors chomping it up. So even if it's there, it goes away so quickly you can't see it. So a lot of learnings. Depending on the test used, though, if the tumor is big enough or quote unquote aggressive enough, ctDNA will typically be found. And we do see it in individuals with primary brain tumors, in other words, brain cancers that started there, or even brain metastases. There's also a parallel effort looking at alternative biofluids. So, blood is obviously where we're looking for circulating tumor DNA, but looking in the urine, looking in cerebrospinal fluid, looking in peritoneal fluid or the fluid in the belly from tumors irritating there, that may be beneficial for certain tumor types as well.
That's an important point. We today are talking about AI dot resistance, circulating tumor DNA, but science doesn't stop. And there's a whole new cadre of potential endpoints coming our way in the future. So it's an exciting time, and it's hopefully a productive, promising time for patient care as well. Lauren, anything to add?
Yes, just kind of sure, thinking about like the future, and I think Hillary hinted at it, but after AI resist, we're thinking about things like looking at total tumor burden and assessing more than 5 lesions, and also going into that 3D space and using AI to help us with automated volumetric analysis of all the total lesions in a tumor. And I think all of us are just waiting to dig into those types of data because I think it will give us a more whole body or even whole organ approach of the disease compared to what we can do now with resist. So really exciting times.
Very good. And to the audience, we love questions, so please feel free to pile on. We have one here that I think is an excellent question for the two of you together. So the team at Friends has picked all the right panelists for today. I am reminded that ctDNA is a great global signal and may someday replace periodic imaging. Can you provide your thoughts on how it will account for disease location and organ-specific response? So a couple of aspects there. Will it in fact replace periodic imaging? I think is a question we might want to delve into. But what can it tell us about the originating tumor?
I don't think ctDNA is going to completely replace imaging. The hope is that it will help gate imaging, perhaps. In other words, instead of doing scans every 2 or 3 months or 6 months because someone said you should, that there's a cadence for an individual patient. This is the right time to peek under the covers. So that's how I view it for timing. The goal would be to reduce the number of images that are needed, but do what I call for-cause imaging. We take pictures if patients have symptoms or an abnormal blood test. This would be a highly sensitive and hopefully specific blood test to say you really need to look now, or things are getting worse. You need to reassess now, or things are great. You don't really need to keep, save time in scans, time away from work, time away from family, if a blood test is clean, normal, if you will. There are, so on one hand, having ctDNA, thinking about the blood as sort of a cauldron where all bad things might go, and then to be able to measure that is great as a global marker. But as we get closer and closer to, and we are, one could argue, in the era of using these tests in the setting of no evidence of disease. So scans are normal, patient had early stage disease, they had surgery, everything is gone. Surgeons love to say that. And we're treating something microscopic. Sometimes knowing where it's coming from would be very helpful for that for-cause imaging. Where do you really need to look, or does it have bearing for management? So in development for circulating tumor DNA are possible signatures, if you will, looking at the ctDNA and that breakdown to tell us where it's coming from, to clue us into the organ that might be involved. This can also have relevance for side effects, predicting side effects from treatment as well. Whole new science coming up.
Coming up. It's coming up. Lauren, what do you think of this question of might it someday replace imaging? I think we heard Minetta strongly state that it will help guide when and perhaps which kind of imaging to undertake. What are your thoughts?
Yes, I fully agree with Minetta that I think ctDNA is a great guideline for reducing the number of images that we may need in a given patient or in a given study. But I do think that they're measuring different scales of disease, right? The ctDNA is picking up this microscopic disease. The images are giving us very specific spatial information, and within the pixel intensities and their spatial representation and their patterns, we can even, understand things about the tumor microenvironment and the genotype of the tumor that we may not necessarily have that same spatial information with the ctDNA, even if we have the genotype itself. So yes, I think they're very complementary.
Very good. Thank you. We've had a new question come in. Since ctDNA has become a great signal, could you please provide your thoughts on minimum residual disease? Is MRD still helpful relative to PFS or OS? So I think that's a big question about the concept of MRD relative to things that are really entrenched like PFS and OS. And I wonder, Minetta, if I could ask you to take that one.
Sure. It's also, some would argue semantics, but I think definitions are very important. So minimal residual disease really came out of blood cancers, right? So the cancer's in the blood, and you can pick up those minute amounts with tests, including ctDNA, that are representative of the cancer in the blood. So that's truly minimal residual disease. When we think about it in solid tumors, where the tumor some elements of the tumor may end up in the blood, but the tumor itself is not in the blood, right? We're talking about ctDNA as a marker of molecular residual disease because it's picking up the remnants, if you will, in the bloodstream. And so that's part of the difference, I think. For defining disease-free survival, progression-free survival, and I think the question is more in a patient who has imaging evidence of disease, and you're looking at maybe tiny little pieces of it, right? Tiny little masses in whatever organ, too small to measure reliably. Things look great. Is ctDNA going to add anything there or not? And again, when you're looking at such a microscopic level, and if you think again of the blood as sort of this cauldron, it's picking those signals up from wherever they may be. So I see it's back to the imaging and the ctDNA complementarity that you really do need both. There will be situations where it's not 2 thumbs up and 2 thumbs down, that there will be discordances. And it'll be interesting to see as imaging modalities improve and AI enhances our ability to interpret those images, how much ctDNA will add to that. I think it will still be complementary. It's just that the balance may shift a little bit. I don't know. Lauren may know better.
Not yet. Maybe after our phase 2 of AI Resist.
Look forward to that.
Well, I'd like to thank you both. We have no further questions from the audience. So thank you both for your expertise, the clarity of thought, your work and your passion in advancing care for cancer patients, and for the discussion today. Thank you for all of that. And I'll hand back to Hillary.
Yeah, and just want to echo what Naveen had said, to thank you both, and also thank you, Naveen, for moderating effortlessly again for us. Thank you all for joining us. Just a reminder to check out our upcoming events that are happening. Feel free to rewatch the webinar or the meeting from February 5th. We also on our website have some more information about some of the advocacy initiatives that we have ongoing at Friends. And thank you all so much for joining us today. We really appreciate it, and look forward to seeing you at our next event.